Okay, we are now going to move on in our study of biological molecules, and I had mentioned earlier when talking about proteins that there are very special proteins that we'd be coming back to, and those proteins are called enzymes. Okay, so as we go through here are the things I want you to be able to do when we are finished studying enzymes. You should be able to identify the primary function of enzymes in all living things. You should be able to explain what is called the lock and key hypothesis in relationship to enzyme function and how they work. You should also be able to explain how factors such as concentration of the enzyme and substrate, pH, temperature, and surface area all affect enzyme function. So let's get right to it and let's talk about what enzymes are. Enzymes, as I mentioned, are a very special type of protein that act as catalysts. So what is a catalyst? A catalyst is a special chemical, in this case we're talking about a special type of protein, that will increase the rate of a chemical reaction. Okay, now our body is continually carrying out chemical reactions throughout the day, our entire life. This is what we call our metabolism. Okay, now without enzymes present, we could not carry out these reactions fast enough in order for us to survive. This is what makes enzymes so important. We could not survive without them. Okay, so let's look at this. How do they speed up chemical reactions? What they do is they reduce what is called the activation energy of the reaction. Every chemical reaction requires just a little bit of energy to get started. That little bit of energy that is needed to start a chemical reaction is called the activation energy. Now, if we look at this graph, we can see what happens if we do not have a catalyst or an enzyme, and when we do have a catalyst or enzyme. The red line here is showing you that in order to get this particular chemical reaction, these reactants here, in order for them to react and produce these products, we need all of this energy, a lot of energy, just to get the process started. But if we throw in the catalyst, or if we add the enzyme into the procedure, the amount of energy drops dramatically. Now we can get these reactions to take place easier and, even more important, more quickly, so that we get enough of the products we need throughout the day, every day. Okay, so again, they simply lower amount of activation energy, which in turn speeds up the processes that allows our metabolism to provide for all the necessary materials. Now, how do they do that? That is what we call the lock and key hypothesis. The enzymes and the substrate, and the substrate is the stuff that's getting broken down or built up, whatever it is, okay, is what's at work here. In this case, every lock, as you know, has a very specifically shaped key that fits into only that lock. The same thing works here with substrates and enzymes. So this substrate right here, and this happens to be a molecule of sucrose, a disaccharide, table sugar, is the key, and it will fit into this particular enzyme, the lock. Okay, and that is due to the shape of the sucrose molecules. You can see the two carbon rings here, and what is called the active site of the enzyme. Okay, so each enzyme has a very specifically shaped active site, and that active site will only allow a certain shaped substrate to enter and attach itself. Okay, so every enzyme is specific, okay, to each type of substrate. There is only one enzyme that will break down this molecule of sucrose. So the two bind together based on their shape. Okay, the chemical reaction can begin to happen because now we have less energy required. The glucose and the fructose get broken down during this chemical reaction. They get released. So now we have some glucose available for cellular respiration. And then when all is said and done, the neat thing about enzymes is that they can be reused. These two monosaccharides get released, and this enzyme will go looking for another molecule of sucrose. Okay, and it just keeps reusing the same enzymes over and over. Okay, so again, we have the key, the substrate, we have the lock here, which is the enzyme. You put the key into the lock, the chemical reaction can occur. We break down our components, we get our two new products, and then we reuse the enzyme. Again, that is called the lock and key hypothesis. Now, there are several factors here that you can see, or that I'm going to go through, that affect how well enzymes work, or the rate of activity. And I have a couple pictured here on each slide. The first one I'm going to look at here for you is the concentration of the enzyme. Okay, the concentration of the enzyme. Okay, so we have our time here. Over a period, here is our product. Okay, a measure of how much product is getting made. If we look at this, with no enzyme, we are producing very little product over a long period of time. Okay, if we add in a certain amount of enzymes, you can see the amount of product goes up quite a bit. It's much higher. If we double the amount of enzymes, we even get a lot more of this. So the concentration is simply the amount of enzymes. So by increasing or doubling our enzymes, or doubling our concentration of enzymes, you can see how much more product we can get. Okay, if we were talking again about the sucrose, we're going to have much more glucose available now for cellular respiration. So the higher the concentration of enzyme, the better the activity, okay, and the more products we are going to get produced. The second thing that can affect it is what we call the concentration of the substrate. In our case earlier, it was the sucrose that is the substrate. So again, we have two lines here, the blue one and the red one. Okay, the blue one is a low amount of sucrose. If we raise the amount of sucrose, we put more into our bodies here, we can see that the rate of reaction will increase. Okay, we have more stuff to break down. So if we increase the concentration of the substrate, we make more available to our bodies, we can obviously get more product. We're going to have more reactions taking place in a shorter amount of time. So concentration of enzyme and concentration of substrate are directly or do directly affect the rate of the chemical reactions. And that means that if we increase the concentrations of the enzyme substrate, we're going to get faster reactions and we're going to get more product. Okay. Next, we're going to look at something called pH. pH, how acidic is something. pH is a measure of how acidic is, and our bodies run slightly acidic. Okay, so again, pH. Enzymes have very specific pH ranges. Each different type of enzyme in your body will work at a very specific pH. That's why we have some pHs that function well in your mouth and some that don't. And on the other hand of that, we have enzymes that don't work as well in the mouth but work very well in the stomach. Okay, because our stomach is very acidic. It has in it hydrochloric acid, a very, very strong acid. Okay, so we have a very low pH in our stomach, while we have a much higher pH in our mouth. Okay, so since these enzymes are specific to a certain pH range, they're only going to work within that range. Okay, so if a change in the pH will reduce the rate at which the enzyme works. So if for some reasons our mouth would become highly acidic, okay, if you eat a lot of sour stuff, that will make your mouth acidic. Okay, many of the enzymes in your mouth that are in your saliva will not work as well. Okay, on the other hand, once we swallow food, there are different enzymes in our stomach that work at that very high, but if we were to take something like an antacid, okay, and reduce that pH range, then again our stomach enzymes would not work it well. Okay, so enzymes function to a very specific pH, and that pH is determined by the type of enzyme. And there are enzymes that work on carbohydrates, there are enzymes that work specifically on proteins, enzymes that work specifically on lipids when we go into breaking down food. Another factor is temperature. Okay, temperature. And we can look here at our graph here and kind of see what's happening. And what this graph is showing us, as you increase the temperature, increasing the temperature is going to add more energy. More energy means more reactions. Remember, we talked about that activation energy. The higher the temperature, more energy is available to keep our chemical reactions take place. But we get to a certain point up here where if we add too much heat, suddenly the enzyme quits working and drops off. Okay, we go all the way back down here to 0. Why is that? Well, enzymes, as we mentioned, are types of proteins. If you raise the temperature too much, the protein will change its shape. Proteins are very susceptible to high temperatures. If a protein changes its shape, okay, you have to go back to your lock and key hypothesis. If the substrate changes its shape, it's no longer going to fit into the enzyme. Okay, or vice versa, I should say. If the enzyme, the protein, changes its shape, it's no longer going to be able to attach itself to the substrate, and therefore the chemical reactions slow down completely. Okay, so if we change the shape of our enzymes where they will not fit the substrate, we will not get any help with the enzymes, and our metabolism slows down dramatically. So temperature is a very important function. Okay. The last thing I want to talk about here is the presence of inhibitors. An inhibitor is a molecule that binds to the active site of the enzyme and blocks access to the enzyme, meaning that the substrate cannot fit into the enzyme and get broken down. And you can see here in the picture, okay, what is happening. Right here is our substrate. It goes through the process, it attaches itself to the enzyme, it gets broken up into its simpler parts and gets released. The problem with inhibitors, they attach to the enzyme and never get released. So they block that enzyme. That enzyme is now unable to function. So we're basically reducing the concentration of the enzyme or functional enzymes, and therefore we cannot get our reactions done as well as we did before. Now, hopefully this little video will pull up in there and we can see what's happening here.
Meet our chemist, Harriet. She has a chemical reaction that needs to occur more quickly. Our chemist has some processes at her disposal that can help her speed up her reaction, and she knows of 5 ways. And to remember them, she thinks back to her days as a high school student